Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Shrivastav. Today our study topic is Maize hybrid development breeding outlook, about which we will discuss. So let's proceed. Maize crop evolved from its wild relative Teosinte about 9,000 years ago in southern Mexico. Maize is a highly cross-pollinated crop which is having separate male which is known as tassel and female which known as ear or cob, and these are the flowers on the same plant, because of which it is called monoecious plant. Maize is highly cross-pollinated with high genetic variability, where the pollination mainly takes place by wind, and there is high outcrossing or high natural cross-pollination more than 95%. Hybrid crop development is the process of crossing two genetically different but compatible parents plants to produce offsprings or hybrids with superior qualities compared to both parents. This hybridization process makes use of heterosis, means hybrid vigor, where the hybrid performs better in yield, resilience and quality than in their parents. Maize is having the reproductive structures which is called tassel, ear or seeds, where the tassel is the male inflorescence located at the top of the plant and consists of numerous slender branches bearing tiny flowers with anthers and pollens, where the ear or cob is the female inflorescence typically found lower on the stem and is a compact spike with numerous florets enclosed within a husk, and each floret contains a silk which is the style stigma for pollination. And the seeds are the kernels we consume are the fruits or grains of the maize plant containing the embryo, endosperm and the pericarp. In case of corn, there is about 90% hybridization, where the seed utilization trend shows 90% of the farmers go with seed replacement by purchasing new seed. And the varietal preference is only 10%, where the farmers use their own source for cultivating the F2 crop or commercial crop. There are different hybrids in maize like single cross hybrids, three-way cross hybrids, double cross hybrids, modified single cross hybrid, double modified single cross hybrids, modified three-way cross hybrids and top cross hybrids, etc. If we see the hybrid segments, it can be divided in two categories like hybrid type area segmentation and second season area segmentation. Single cross hybrids are having the high yield potential. There are input responsive, uniform, and single cross hybrids are almost contributing 55 to 60% of hybrid market. As three-way cross hybrids are concerned, they are more stable, having the moderate to high yield, and they are cheaper compared to single cross hybrids and almost having the contribution of 20 to 25% of hybrid market. And double cross hybrids are old hybrids which are rarely used now, and they are persisting in older market. And as the specialty hybrids are concerned, there are sweet corn, quality protein maize hybrids and popcorns which are having the participation of 10 to 15% of total hybrid market. As seasonal cultivation is concerned, maize or corn is grown in three seasons: Kharif, which is also known as wet season, where the Kharif crop contributes about 75% of total corn cultivation, and Kharif is the monsoon crop, where the crop is prone to pest and diseases, and these are grown in Bihar, Uttar Pradesh, Madhya Pradesh, Rajasthan, Gujarat, Karnataka, Andhra Pradesh, Telangana and other states. Second season is Rabi, which is also known as dry season crops, which contributes about 21% of total maize cultivation, and this season grown crops are having the high productivity. They are grown in irrigated belts like Andhra Pradesh, Telangana, Karnataka and Bihar. Third season is spring or summer season crop, which is having the very negligible 4% contribution of maize cultivation, and these hybrids are having the nature of short duration, and these are grown in eastern India like Bihar and UP, and in Punjab, where spring crop is having the high seed demand. As an economic importance, maize is one of the world's most important cereal crops, providing a stable food for millions of people. It is a major component of livestock feed, contributing to meat, dairy, and egg production. And as biofuel production is concerned, maize is used to produce ethanol and a biofuel that can replace fossil fuels. Maize is used in various industries, including food processing, pharmaceuticals, and bioplastics. So, in India, the corn seed industry is growing, and there is area shifting from other crops to corn, and there is great demand for poultry and animal along with industrial usage, food industry, ethanol, and other miscellaneous usage. In India, if we see, the commercial market is around 2 lakh metric tons, where it is grown in Kharif, Rabi, and Spring. Kharif is contributing about 70% of the total market, Rabi is contributing around 23%, and Spring is having the 7% of contribution. If we see the commercial cultivation area potential, maize is grown almost in 12 million hectares area, out of which Kharif is having 9 million hectares area with contribution of 75%, Rabi is having the cultivation of 2.5 million hectares with 21%, and Spring maize is having the 0.5 million hectares with only 4% of cultivable area. If we see the hybrid corn seed production, approximate 40% production and supply is done by the domestic companies, and MNCs are contributing around 60% production supply to the farmers to meet the demand. Now we will go through corn breeding research. Long-term breeding effects in corn is the blend of progress and pressure. There are integrative genomic, physiological, and epigenetic research, and the goal is that next generation of hybrids remains productive, diverse, and resilient. And the long-term selection in corn breeding is resulting in delivering high yields. This shaped the genetic, physiological, and epigenetic foundation of modern hybrids, and understanding these vitals for future sustainability. As the goals are there to develop the high potential sustainable hybrids, and here we will reveal how the decades of breeding have influenced genetic diversity, adaptability, and performance. A genetic trend over time is concerned, it has given the genetic diversity, heterotic structure, and allele frequency shifts. As the physiological and morphological shifts are concerned, there are shorter duration, stronger stalks, upright leaves or erect leaves plants, better light interception, improved the stay green characteristics, and dry down higher density tolerances. And epigenetic and transgenerational effects has resulted in high yielding stabilities, stress response, and hybrid vigor, where the gene and environment and climate resilience has given the combined productivity with broad adaptability. And there are innovative research methods which are the integrative technologies concerning the high through output phenotyping, genomics, transcriptomics. After that, epigenomics, machine learning based predictive models, and time series data analytics, etc. Maize breeding combines classical methods like selection, inbreeds, hybrids with modern molecular genomic tools with ultimate aim of high yielding, resilient, nutritionally rich, stable hybrids and varieties which are suitable for farmers' needs. As the commercial corn breeding is concerned, it is the intersection between advanced genetic science and the biological, environmental, and economic stresses which are known as strain that define modern agriculture. And commercial corn breeding reflects is not just about producing hybrids, it's about balancing scientific innovations with real world pressures. And the future corn breeding directions are developing strategies for sustainable gain, optimal contribution selection of parental lines and hybrids, germplasm diversification, and there should be blend of tropical and temperate, epigenetic informed breeding, climate smart edutypes designs for suitability to the various geographies, and the outcome should be resilient hybrids that sustain yield and protect diversity. Corn breeding is the intersection between science and stress. As the science is concerned, it refers to the genetics and biotechnology, where the use of molecular markers, genomic selection, gene editing, and double haploid hybrids are done to accelerate genetic gain. The quantitative breeding principles refers to the heritability estimations combining ability, gene and environment expressions, and analysis, where the data-driven breeding refers to the use of artificial intelligence, phenomics, and predictive modeling for faster and more precise hybrid development. And the seed system science ensures in uniformity, vigor, and seed quality at commercial scale. Where the stress is concerned, it refers to the biotic stresses, abiotic stresses, socio-economic strain, and environmental strains. Where the biotic stress refers to the pest, disease, and pathogen resistance. Abiotic stresses refers to the climatic like drought, heat, nutrient deficiency, salinity, and increasingly critical under climate change. Socio-economic strains having the trend of market volatility, a smallholder affordability, and regulatory hurdles to be taken care. And as the environmental strains are there, it refers to the soil degradation, declining input use efficiency, and sustainability pressures. If we see, each strain or line of population carries a unique genetic background, and the success of breeding lines in identifying complementary strains that maximize heterosis means hybrid vigor. Commercial corn breeding continuously refines and recombines these strains to produce superior hybrids. Corn breeding is the combination where art meets science. Science provides the foundation and art refines the outcome. The best hybrids are born from the synergy of both. Science refers to the precision, prediction, and measurable progress, where the arts in the breeding meets with creativity, institution, and human judgment. Corn breeding is deeply rooted in science, which refers to the genetics, statistics, and molecular biology. And the science involves genetic inheritance, which refers to the traits like yield, maturity, and disease resistance from old generation to the next. And the quantitative genetics refers to the estimating heritability, combining ability, and genetic variance components. And the molecular tools refers to the use of DNA markers, genomic selections, and CRISPR based gene editing to accelerate improvement process. And data driven decisions are including field phenotyping, genomic data, and artificial intelligence tools models to help identify superior hybrids faster. Where the art is concerned, despite scientific tools, corn breeding still requires a breeding intuition and vision. And there should be recognition of plant architecture, ear traits, and stress responsive by the eye in the field. And making strategic crosses based on experience and instinct, not just data, and balancing adaptation and performance for specific agroclimatics, and understanding aesthetic traits like kernel color, ear uniformity, and grain texture. So, by applying the science and art, we can get the best hybrids. For hybrid maize product development, the germplasms are required based on agronomic requirements, abiotic stresses to meet biotic stress and nutritional qualities. Agronomic requirement refers to the germplasm can be fastened and meet to the fast track. It should have high yielding potential, plant density suitability, lodging resistance, and enhancing resource use efficiency like nitrogen use efficiency, water use efficiency, and carbon use efficiency for improving photosynthesis and yielding potential. And the germplasm should meet the abiotic stresses, which refers to the climatic like drought tolerance, heat tolerance, cold tolerance, waterlogging tolerance, and stay green traits to fit in different geographies. And the germplasm should having the potential to fit to the pest and disease tolerance like fall armyworms in maize. And nutrition quality refers to the storage quality, kernel color, and usage in food like sweet corn, baby corn, and biofortification. And quality protein maize should have high lysine or tryptophan. So these are the requirements for developing the lines for future requirements. Here we will discuss about the featured modern plant traits which are required for corn breeding for future sustainability, and it refers to the plant height, leaf orientation, ear placement, tassel size, root system, stalk strength, flowering synchrony, stay green traits, growth habits, density tolerance, canopy temperature control, and stress adaptations. So these plant architecture and features should consist to sustain to the market for developing the best suitable hybrids in different conditions. Now to discuss genetic control of different characteristics or traits in maize. There are three traits, like first one is plant aspect traits, second is ear traits, and third one is grain traits. As plant aspect traits are concerned, first one is highly heritable, which are mostly additive gene action traits like plant height, ear height, leaf angle, leaf architecture, ASI, stalk diameter, tillering, and plant type, architecture, tassels, etc., which decides the productivity of the during the production. And second was low moderate heritable traits, which are non-additive gene action, and such traits consist of lodging resistance, response under drought stress, and pest and disease tolerance. Ear aspect traits refers to the predominantly additive gene actions like ear length, ear girth, kernel rows per ear, cob diameter, husk cover, and second one is mostly non-additive gene action traits, which refers to the ear weight, number of ears per plant, selling percent, resistance to ear rot and earworms, and uniformity of ears. And the grain kernel traits are controlled both by additive gene and non-additive gene. As the additive gene action is concerned, they are high to moderate susceptibility, and they are easy to fix, and there are like kernel type, endosperm, kernel color, kernel length, kernel breadth, and seed test weight, and nutrient contents refers. While the strong non-additive gene of ear grain traits concerns are there, it refers to the grain yield per plant, rate of dry down kernel moisture at harvest, and grain appearance and uniformity. Here we will discuss about key parameters for a strong yield potential in hybrid maize. The collected data helps in accurate selection and supports to the identification of lines with a strong yield potential, and the key parameters which contributes to the strong yield potential like selling percent, number of grains per row, number of rows per cob, anthesis interval, seed setting ratio, 1000 grain weights means test weight, cob length, cob girth, plant height, ear height, lodging resistance, disease response, maturity time, plant vigor, and kernel type and color expression. These parameters give a clear picture of each hybrid and need to compare entries in real field conditions to find lines that show consistent performance, a strong seed set, and balanced plant architecture. This evaluation supports advanced decisions and improves future product development. Maize physiology is very important for achieving higher productivity. And maize productivity physiology refers to the different parameters of physiology like photosynthetic efficiency, which relates to the biomass and kernel weight, Second one is water use efficiency, which refers to the stable yield under low water conditions. Third one is nitrogen use efficiency, where high yield should attain even at moderate nitrogen inputs. Fourth one is radiation use efficiency, where the dry matter should result into the grain yield. And the source sink balance, which refers to the fast grain filling rate by high carbohydrate transfer to the grains. Fifth one is root shoot coordination, where the balanced vegetative growth refers and compared to the higher harvest index. And stress physiology, where the crop should be protected and till the reproductive stage and yield. So final outcome refers to the higher kernel number, higher grain weight, better stability and higher total productivity. Here we will discuss about maize hybrid seasonal segment requirements. For Kharif season, the maize hybrid should have early maturity, medium to tall, tolerant to lodging, tolerant to drought conditions, and fit to the relevant condition from 600 to 800 mm. And these Kharifs are mainly grown in Karnataka, where the crops should have tolerant to fall armyworms and fitted to the mechanization suitability. They should have high test weight, good grain quality, tolerant to stalk rot, and high yield expectations. And generally, the flint seed is more preference and no dent seed preferred here. And the color should have a specific yellowish to orange. In Rabi, there are different preferences like long duration, good extendability, high input management condition. Hybrid should be responsive and it should have adaptations to high plant density, lodging tolerance, high selling percent, because the Rabi yields are more than Kharif and it is more having expensive cultivation practices. And the Rabi is mostly grown especially in Bihar, where the hybrid should fit to the temperature range 25 to 30 degrees centigrade, tolerant to cold and heat, and high yield expectations, and stray green characteristics at harvest, which are suitable for fodder trade. As spring hybrids are concerned, these spring hybrids are used in replacement of pulses crop and sunflower, and the hybrid should have tolerant to heat and having the early to medium maturity preferences. They should have excellent extendability quality and high management responsive, high selling percent, and fast drying down, high yield expectations, and fit to the respective areas crop rotations. And spring hybrids are grown in north, where the hybrids are mostly temperate corns, and the flowering falls in April, where the temperature is running 45 degrees centigrade. So hybrid should have well suited to the high temperature. While maize line development, key traits are required to maintain the sustainability. In case of Kharif season, the hybrid preferences are like high yield potential, heat and drought tolerance, and hybrid line development, which are against post flowering stalk rot, bacterial leaf sheath blight disease, and rust diseases. And the grain type should be flint to semi-flint, dent and semi-dents preferred, and good extendability with husk cover to protect from rains. In case of Rabi, the lines requirement preferred like high yield potential, cold and heat tolerance, and erect plant type, which suited to the high density planting and having good extendability and high test weight. As the spring hybrids are concerned, they should have high yield potential, cold and heat tolerance. They are mostly preferred like erect plant, which suits to the high density planting, having good extendability, high selling percent or recovery, because of which they should get high prices, and 20% tropical characteristics and 80% temperate characteristics blending. So these are the specific requirements of different cultivation season segments. This slide speaks about the breeding objectives, where the breeding goals refers to the developing pure stable varieties or hybrids with high yielding potential, maintaining variability and exploiting heterosis, and the hybrid should fit to the abiotic stasis, and meet to the nutritional requirements. And there are different breeding methods used like mass selection, recurrent selection, inbred line development, hybrid breeding, and controlled pollination is essential in breeding activities. Maize is a cross-pollinated crop and it tends to be heterozygous and heterogeneous, and continuous variation is required to maintain in the population. And there is requirement of driving genetic diversity where the increasing size of breeding program needs for greater selection intensity and enhancing the breeding cycles, and more accurate selections are preferred for higher heritability, and infusing new and useful genetic variations and use of diploid diploid hybrids, double haploid fast inbred lines for increasing speeding the breeding cycles. And there are breeding operations because maize is a heterotic crop, so parental line selection is per the breeding objectives and focus given to the elite and disease resistance lines and accurately implementation of the programs. Here we will discuss about different germplasms of maize hybrids. There are three germplasms like tropical germplasm, subtropical germplasm, and temperate germplasms. For tropical germplasms, they are adapted to hot and humid climate with the little temperature fluctuations, and the key characteristics are defined as temperature tolerance, which are high heat tolerance and poor cold tolerance, and the growth duration is generally long duration, and they are having the a strong photo period sensitivity, means they are having the long days, and the plant architecture is generally tall, leafy, large tassels and late maturity, and the kernels are generally flinty endosperm, common and high genetic diversity, and they are having the disease resistance of broad spectrum, tolerant to tropical diseases and pests, and they are often derived from tropical populations, and these lines are used for breeding for tropical regions. As subtropical germplasm is concerned, they are adapted to moderate climates with distinct seasons, and as key characteristics are concerned, they are moderate and better cold tolerance than tropical types, and they are medium duration hybrids lines, and they are having the intermediate photoperiod sensitivity, and as a plant architecture is concerned, they are medium height and better lodging resistance, and the kernel traits are mix of flint and dent and good grain filling under moderate stress conditions. And as a disease resistance is concerned, they are balanced disease resistance and adapted to both tropical and temperate stresses, and they are having the heterotic group of intermediate between tropical and temperate. And these subtropical lines germplasms are used for developing hybrids for warm but not equatorial zones, and like Indo-Gangetic plains and middle latitude of Africa and Latin America. And the third one is temperate germplasm, which is adapted to cooler temperate climates with long summer days. And as a key traits characteristics are concerned, they are having the high cold tolerance and poor heat tolerance, and they are short to medium duration germplasms and having the very low photoperiod sensitivity, and the plant architecture is shorter, compact, having the erect leaves and efficient photosynthesis capacity, and the as a kernel traits are concerned, they are mostly dent type and having the high yield potential, and as a disease resistance, they are adapted to temperate pathogens like stalk rot, and they are heterotic groups like corn belt in having the yellow dent, and they are high yielding commercial hybrids and used for temperate regions. The key challenges related to corn hybrid breeding is germplasm diversity maintenance because of heterotic groups, and balancing for yield versus abiotic and biotic stresses, and developing tolerant material, and having limited testing locations, and a review of data computing analysis and developing the strategies. The crop dynamic changes needs for continuing improvements like ensuring full utilization of germplasm technologies and trialing and networks experts to train and implement modern technologies, staggering like preference of low staggering hybrids, same day sowings. No sensitive lines are preferred and to the market, and they should have good storability, and working with multicultural team for the success, and annual review meetings for find out the different constraints and new strategy development. For quality maize breeding, there are process for developing high lysine tryptophan hybrid lines, nutrient enriched maize which are having the content in the zinc, iron, provitamin A, and climate resilient maize development tolerant to drought and heat, and bioenergy maize for ethanol usage and silage use. And there are impact on maize breeding because of shorter breeding cycles, more precise trait requirements, and better adaptation to climatic change condition, and there is further requirement for increasing yield quality and resistance to drought and pests and other climatic, biotic and abiotic stresses. Here to study about exploiting maize diversity. The foundation of breeding programs depends on maintaining broad genetic bases through landraces, synthetics, and wild relatives, and by applying hybridization, recurrent selection, and molecular tools. And there is the development of hybrids and varieties with higher yield, resilience, and nutritional quality. And the methods of exploiting diversity used as a hybrid breeding heterosis and cross genetically diverse lines like crossing between dent and flint, tropical and temperate lines. And the sources of maize diversity are depends on land races which are rich in genetic variability and adapted to local environments, and the wild relatives like Teosinte tripsecum which provides resistance genes for disease and stresses, and the exotic germplasm usage which are useful for broadening the genetic base, and elite inbreds usage which are already improved but having the limited variability, and the synthetic and composite populations which are required to maintain broad variability for selection. Now to discuss about maize trait fixing. There are some traits which are easy to fix because they are controlled by monogenic lines and they are having the qualitative characteristics, and such traits consist of kernel color, kernel textures, pigmentation, male sterility, fertility restoration, and single gene disease resistance. While there are some traits which are having moderate to fix, like these are controlled by oligogenic and having the medium heritability, and such traits like flowering traits, ear rot resistance, husk cover, cob traits, green characteristics, stalk strength, and lodging resistance are falling under these traits. And there are some traits which are difficult to fix, and these are quantitative characteristics and controlled by polygenic. So these characteristics can be defined as grain yield, grain quality, plant height, ear placement, ear size, and kernel row numbers, maturity, stress tolerance, and complex disease resistance. In hybrid maize, single cross hybrids are leading and they contribute around 60% of total market segment. So the best suitable lines development is very important, and in such cases for the making the effectiveness in HSP hybrid seed production cost, there is need to diversify the production environment geographies and use of male sterility system. In single cross hybrid, females should have high yield potential and female seed size should be small to medium size of kernels, and these lines should have better and good adaptability in high density planting systems, and male should have bold kernels, and these lines are having the flint seed where the hairpin cracks are observed and it may impact for shelf life. So we have to think carefully about making the operations to protect the shelf life of the product. And the single cross hybrid should be suited to mechanization for detasseling and harvesting, and breeder have to develop the lines to be used for same day staggering or low staggering hybrids, and these lines should have no sensitivity for production conditions. Now to discuss about maize breeding techniques. First one is marker assisted selection, MAS, which refers to the use of molecular markers linked to desired traits to aid in selection. And this MAS is used for the selection for disease resistance like triticum leaf blight, downy mildew, and improvement of quality traits like provitamin A, lysine and tryptophan, quality protein match, and it reduces the breeding cycles and increases selection accuracy. Second one is genomic selection, which uses genome-wide markers and a statistical model to predict breeding value without phenotyping every generation. And the applications are use of complex traits like grain yield, drought tolerance, and stalk strength, and it reduces cost and time of field evaluations and enables early generation selection. Third one is genome editing, which refers to the precise modification of DNA at specific loci without introducing foreign DNA. And as applications are concerned, it is used for enhancing drought tolerance and developing male steady lines for hybrid breeding and removing undesirable alleles from elite lines. Fourth one is transgenic approaches for developing GMH, which refers to the introduction of foreign genes for specific traits like insect resistance, Bt maize expressing Cry1AB, Cry2AB genes, and herbicide tolerance, glyphosate resistance maize, and stack trait for multi-stress resistance. Fifth one is double haploid DH technology, where the production of complete homozygous lines in one generation using haploid induction and chromosome doubling. And the applications are for rapid development of pure lines, which enhances hybrid breeding efficiency and saves 4 to 6 years compared to the conventional methods. Sixth one is genotyping by sequencing and SNP genotyping, which refers to the sequencing technology for high resolution genotyping, and it is for the construction of high density genetic maps and identification of quantitative traits and diversify analysis and population structure studies. There is a speed breeding, which refers to the controlled environment to grow multiple generations per year, and it reduces time need for inbred line development and ideal for trait introgression and backcrossing programs. Eighth is multi-omics integrations like genomics, transcriptomics, and proteomics, metabolomics, which are used for understanding complex trait regulation like trait response, yield, and discovery of gene networks for targeted improvement and fine-tuning of breeding strategies. Now here we will discuss about double haploid technology in maize. Double haploid breeding is a technique used to produce completely homozygous lines in just 2 generations instead of 6 to 8 generations of conventional breeding. Double haploids are plants that have 2 identical set of chromosomes, making them completely homozygous, and they are produced through a process that starts from a haploid cells with 1 set of chromosomes and then double the chromosome number. And double haploids plant created by doubling the chromosomes of a haploid cells, resulting in a diploid homozygous plant. Haploids are produced naturally in maize, and they were first identified in the crop about a century ago, and today they are used widely in different breeding programs, particularly in the development of double haploids, which are highly uniform, genetically pure and stable. Double haploid technology has simplified logistics to make the maize breeding process more efficient and intuitive, facilitated strategies at the molecular and genomic level, and increased genetic gains in different breeding programs. As a DH breeding role is concerned, there is rapid development of pure parental lines. DH technology significantly accelerates the breeding cycle, allowing for faster development. And there is early hybrid testing and selection, commercialization of new inbred lines and hybrid varieties. And DH breeding facilitates marker assisted and genomic selections, enhances breeding efficiency, which helps in quickly incorporating desirable agronomic traits like drought tolerance into elite germplasm and boosting genetic gains in cost effectiveness. And as research and applications are concerned, DH lines are available for basic research such as mapping genetic regions for specific traits and for applied breeding, enabling the rapid creation of pure lines. As the DH breeding is concerned, the stepwise process referred to the haploid induction and pollination using a haploid inducer line which produces haploid embroids. And second one is haploid identification using genetic markers or phenotypic traits. And there is chromosome doubling, treating often with colchicine or nitrous oxide to double chromosome and restore fertility. And there is next step is DH live formation resulting plants fully homozygous double haploid and hybrid development where the DH line serves as parent for hybrid seed production. Polygenic traits in maize breeding. Polygenic traits are characteristics controlled by multiple genes and each with a small additive effects. These traits are typically quantitative, influenced by the environment and show continuous variations. And these polygenic traits can be expressed like grain yield per plant, where the primary breeding objective is to increase the yield and it is controlled by many minor genes. Second one is plant height, which affects lodging resistance. Other traits like ear length and kernel rows per year, which influence yield potential. After that, anthesis and silking interval, which is critical for straight tolerance and pollination efficiency. And after that, dot tolerance, which involve root architecture, stomatal regulation, and osmolytic accumulation for resistance. After that, disease resistance, quantitative resistance, which refers to the durable resistance against multiple pathogens, and extendability and stock strength, which prevents lodging and improves harvest efficiency. Now we will go through understanding straight and reciprocal crosses in maize. Straight cross where the A parental line female crosses with B female, but in case of reciprocal crosses the same two parents are crossed reverse, where the parent A becomes parent B and parent becomes parent A. So the practical usage of reciprocal crosses are used for better seed setting and desirable cytoplasm to used like female for commercial habit production, and cytoplasmic male sterility where the reciprocal crosses help detect suitable cytoplasmic background for male sterility system, which is critical in hybrid seed production. And reciprocal crosses are also used for yield optimization where the reciprocal testing ensures that the chosen female parent doesn't depress yield due to unfavorable cytoplasmic or maternal effects. Now to study the combining ability in maize breeding. Combining ability refers to the ability of a parent line, inbred variety or genotype to transmit its desirable traits to its offspring when crossed with another line. It helps breeders to identify the best parental combinations for hybrid development. Combining ability is used in maize breeding and heterosis because maize is highly cross-pollinated crop and exploitation of heterosis is a major objective in hybrid maize breeding. Combining ability is important because it helps in selecting superior parents for hybridization, and it determines whether a crop improvement program should focus on additive or non-additive gene effects, and it is crucial for heterosis breeding because heterosis exploitation leads to high yielding maize hybrids. And combining ability also is useful in identifying heterotic groups and tester lines and used to identify the best parental combinations. Stress combination breeding in maize, which is also known as multiple stress tolerance breeding. Stress combination breeding is the process of developing maize hybrids or inbred lines that can tolerate two or more abiotic or biotic stresses simultaneously. like drought plus heat, or fall armyworm plus nitrogen, or high temperature. The stress combination breeding is important for multiple stress interactions, climate change stress overlapping, and ensure yield stability and resilience. Real world conditions rarely involve just one stress, and climate change is increasing the frequency of combined stresses. And in such conditions, farmers need resilient hybrids that perform consistently across environment. So, for the stress combination breeding, there are need for breeding strategies to develop maize hybrids that tolerate multiple stresses simultaneously, like abiotic and biotic. And the stress combination breeding tools are used as conventional hybrid breeding, marker assisted selection process, genomic selections, double haploid DH technology, and stress phenotyping and multi-location trialing. And the key traits targeted in combination breeding, like root traits for drought escape, stray green traits under heat, silk extrusion and pollen viability under high temperature, fall armyworm resistance genes, stalk strength and disease resistance. Now we will study about stray green traits in maize. Stray green trait is a critical adaptive trait in maize, especially in water limited or stress prone environments. It correlates positively with yield, improves source strength, supports better grain filling, and is increasingly integrated into modern hybrid breeding programs targeting climate resilience and yield stability. If we study stray green characteristic in deep, first one is physiological basis stray green, which involves delayed leaf senescence, particularly during the grain filling period, and the key features include chlorophyll retention, continued photosynthesis, active sourcing activity remains post anthesis. Second one is correlation with yield conditions, where the stray green effect on yield having the positive correlation aids in better extendability grain filling. Drought or heat stress a strong positive correlation due to longer duration of effective photosynthesis. And late season stress protects yield by preventing premature senescence. And it is associated with yield benefits, where the higher kernel weight and grain numbers and better grain filling rates reflects under stress, and the improved biomass percentage to the year and greater harvest index. Third one is genetic and molecular insights of stray green. The stray green is quantitative polygenic yield stability, which refers for the drought resistance, photosynthetic efficiency, and major contributing gene involves chlorophyll degradation. And fourth one is stray green agronomic importance, where the drought tolerance maintains grain filling under terminal droughts, and disease resistance cause delay senescence, which reduce foliar disease impact. If we see the empirical evidence, the CIMIT trials show 10 to 30% higher yields in stray green maize under drought stray, and stray green hybrids show longer grain filling duration by 5 to 10 days. If we see the stray green traits in breeding applications, stray green is a secondary selection trait for drought breeding and climate smart hybrids, where the marker assisted genes are used and genomic selections used, and combining ability of stray green traits evaluate in hybrid parents. There are challenges related to stray green breeding considerations, like too much stray green may delay maturity and harvest, and trade-off with early vigor, which needs balancing for season length, and stress escape versus stray tolerance. Stray green is stray tolerance and not the escape. Now we will discuss about drought resilience corn, where the root architecture should be deep and vigorous. Leaf traits reflects to the erect leaf orientation. Tassel and flowering traits refers a smaller tassel size, and stem and plant architecture shows robust stem with good carbohydrate reserves, and ear traits reflects reduced bareness. Drought resistant corn hybrids at flowering combine deep and efficient root systems, erect and conservative leaves having a smaller tassels, short anthesis silk interval, strong stems and stable ear set. And these traits together ensures better pollination, kernel initiation and yield stability under water limited conditions. Thank you very much. This is the end of my presentation. See you shortly. Thanks.